This research focused on Bifidobacterium, a probiotic bacteria which lives in the colon. Bifidobacterium is a probiotic because it causes anti-inflammatory properties and specifically lives inside the gut. This microbe was chosen specifically because of its abundance within our gut microbe and its early presence and early gut health being correlated to later life health. Additionally, its absence is correlated with certain types of chronic gut illnesses, such as inflammatory bowel disease. We chose to study Bifidobacterium adolescentis, longum, brev, and dentium. Bacteria as a whole have lipoprotein structures, which are proteins that are embedded in the outer membrane, which carry out a variety of functions. One thing that they all have in common, however, is an N-terminal cysteine, which is present in all varieties of lipoproteins. The immune system, and specifically toll-like receptors and toll-like receptor 2s, use this N-terminal cysteine as a way to recognize foreign invaders and then cause later pro-inflammatory responses. This requires TLR2 combining with either TLR1 or TLR6. Bringing this back together, Bifidobacterium lives within the colon where there are epithelial cells which have a lot of these TLR receptors on them. It would be expected that the Bifidobacterium lipoproteins would stimulate the immune system, causing pro-inflammatory responses. However, this isn't seen, thus leading to questions around the immune interactions with the Bifidobacterium. In order to investigate this, we did two experiments, one investigating the cell as a whole, and then another one investigating the specific lipoprotein structure. For the first experiment, human embryonic kidney, or HEK cells, were chosen because they have been pre-engineered to express the TLR2, TLR1, and TLR6 receptors on them. When exposed to varying concentrations of lipoproteins, in this case, a quarter dilution, where each lower row has a quarter of the dilution, they will then release a molecule which, when added to an activator, will cause a color shift. Here we see the color shift happening in the different types of strains used. And specifically, this color shift can be quantified and then related back to the amount of immune stimulation. Bifidobacterium longum and brev had the highest immune stimulation, while adolescentis and dentium had lower stimulations. For the next experiment we conducted was to first isolate the lipoproteins and then separate them by mass. In the lower middle picture, we see on the left a ladder which has different known sizes, and then the lipoproteins separated by mass. The lipoproteins chosen were around the 50 to 70 kilodalton range, a weight, and then Bifidobacterium dentium was not chosen due to its non-straight band. When we performed the mass spectrometry, an experiment which reveals the mass per charge ratio on these bands, and then cross-examined them with expected lipoproteins, we came up with candidate lipoproteins, which we expect to be in the Bifidobacterium cells. Here we have the different types that could be in it, that being either diacylated, lyso, triacylated, or acylated, due to us not knowing which specific one it is, however, that being later testable, as well as C18-0, 18-1, or C16-0. These were chosen due to previous research done showing that these are the most common lengths, C18 being 18 carbons with 0 double bonds. The next steps would be to optimize growth conditions in order to get distinct bands and to perform a mass spectrometry to confirm the candidates' lipoproteins. Overall, this shows us that Bifidobacterium does have an immune response to them as an entire cell, and hopefully when confirming the candidate lipoproteins, we are able to investigate this further to view if it's something to do with the receptor lipoprotein interactions or if it's a different type of interaction which doesn't cause the overall pro-inflammatory responses.